arXiv paper, so it didn't get into any journals. But we might do another version of it too. It's up to you. Because I know that people have done like different comparisons like that. It's not just like, this isn't the only paper on it, but it's just like trying to get like people usually get the same set of issues over and over, and like getting some new issues in there would be good. But all right, nice to see everyone here. Lilith, Soham, Morgan, Dick, hello. Okay, so today I wanted to talk about a couple of papers again.
So the first paper today I'm going to talk about is this paper, Gene Regulatory Patterning Codes and Early Cell-Fate Specification of the C. elegans embryo. This is Allison Cole, Tamar Hashemshoni, Zhuo Du, and Ite Yanai. And so the abstract reads, pattern formation originates during embryogenesis by a series of symmetry-breaking steps throughout an expanding lineage. So this is where we have our lineage tree, and the lineage tree divides into, so one cell divides into two cells divide into 4 cells, and you keep going. Now, throughout the lineage tree, first of all, you get asymmetric divisions. So you get divisions where there's only 1 cell produced instead of 2 from a division event. The other example of a symmetry-breaking step is where cells will divide at different rates. So if we think about our hypothetical lineage tree, we divide from 1 cell to 2 cells to 4 cells to 8 cells to 16 cells. And generally that's the way it works early on in C. elegans development. And then later on in C. elegans development, you start to get not multiples of 2, but you get sort of these rounds of division that are in between multiples of 2. So you might have 2 cells on the end of your 16-cell embryo. On the anterior end, they might undergo a division event, which gives you an 18-cell embryo. And then the rest of the cells divide, which then gives you your 32-cell embryo. So this is, kind of the symmetry-breaking steps. They're divisions that occur sort of asymmetrically. They're asymmetric divisions themselves. And those lead us in C. elegans because the cells are all deterministic and the cell lineage tree is deterministic to the emergence of different structures at different times. So embryogenesis essentially is the symmetry-breaking process as much as it is a pattern formation process. Now, they talk about Drosophila here. The paper is about C. elegans, but they talk about Drosophila a little bit at the beginning. So in Drosophila, classic work has been shown that the embryo segmentation is established by morphogens within a synticum. So a synticum is a single cell with multiple nuclei. So muscle is a synticum. This is multiple nuclei, but a single cell. And so you get this synticum stage in Drosophila development where you get all these nuclei within a structure, and then that structure cellularizes and you get to the next step of development. So this is what they're kind of getting at here. So they're talking about within the synticum, you have the segmentation, you have these morphogens, which are these signaling molecules, and then the action of genes that set up these gradients that produce sort of gradients within that synticum. And you see like striping, or you see other types of gradual change across the anatomy of Drosophila. So from anterior to posterior end of that synticum, you see gradual differences in what's being expressed. So that's in Drosophila, and we'll see images of that later. This classic model, however, does not translate directly to species that lack a synticum, such as C. elegans, where cell fate is specified by cell-autonomous cell lineage programs and their intersignaling. So a lot of the work on Hox genes, on gap genes, on segment polarity, and that sort of thing was done in Drosophila. And that work in Drosophila applies to mammalian systems, but it does not apply to C. elegans. which is a fellow invertebrate, but it doesn't have the same. So we're actually looking in C. elegans here. We're thinking about a different system altogether. We're thinking actually about this kind of system where we have a deterministic lineage tree. So previous single-cell RNA-seq studies in C. elegans have analyzed cells from a mixed suspension of cells for many embryos to study late differentiation stages or individual early-stage embryos to study early gene expression in the embryo. So what they're doing here is they're doing single-cell RNA-seq. They're looking at different cells. They get a mixed suspension of cells, and they're going to sort of do this study where they look at the expression of cells in different sublineages in this lineage tree, and it may tell us something about how the origins of this asymmetry. To study the intermediate stages of early and late gastrulation, 28 to 102 cell stages respectively, missed by these approaches, here we determine the transcriptomes of the 1 to 102 cell stage to identify 119 embryonic cell states during cell fate specification, including equivalence group cell identities. So they're looking at these two stages of development, early and late gastrulation. This is kind of, getting out of this period of doubling, this early period of doubling. When you get to about 16 cells, you get to 16 cells. You have these, actually at 8 cell, you have the founder cells. And then at the 16 cell stage, you have these sublineages that emerge. And so the sublineages are, sometimes they're specialized for different types of cells. Sometimes it's a mix of cells in them. And sometimes it's the germline. So we'll see later that, there are these different sublineages that are already forming. And so now, we know kind of what type of adult cell it's going to be. And we'll see from the lineage tree that their developmental cells have a nomenclature that matches these sort of sublineages in the developmental lineage tree. And so we start to get these sort of asymmetric division events where we get the 28 cell stage, we get 102 cell stage. So you can see that gastrulation is producing, or other processes as well are producing these symmetry-breaking steps. But then we also want to identify different types of cells. So cell fate specification, what is the differentiation state? Is there like an intermediate state that we can identify? We find the gene expression programs are modular according to the subcell lineages, each establishing a set of stripes by combinations of transcription factor gene expression across the anterior-posterior axis. So we know, of course, that the lineage tree is organized anterior to posterior. The left side of the tree is anterior, the right side is posterior. The right side of the tree has the germ cells. The left side of the tree has the A, B sublineages, and everything in between is defined. And so instead of the sort of striping that we see in Drosophila, we just simply see things happening at the anterior end, the posterior end, and sort of in the middle. And so this is kind of an interesting difference between these two types of systems. Now we know that C. elegans shares some of these genes, or at least that they share gap, pair rule, and segment polarity genes. And so we'll see if they play a role in what's going on here. In particular, expression of the homeodomain genes establishes a comprehensive lineage-specific positioning system throughout the embryo, beginning at the 28-cell stage. So we actually get this sort of positional information, or this positional independence of these cells. And so we are already start to see that here, and it involves some of the homeodomain genes we see in the striping in Drosophila. So moreover, we find that genes that segment the entire embryo in Drosophila have orthologs in C. elegans that exhibit sublineage-specific expression. These results suggest that the C. elegans embryo is patterned by a juxtaposition of distinct lineage specification genes, regulatory programs, each with a unique encoding of cell location and fate. So they start talking about developmental toolkit genes, which is sort of what we have in sort of determining the patterning of the organism, how patterns are formed, or how we go from these undifferentiated cells to the sort of these patterns or these differences within the embryo. And so we can think of that as patterning, we can think of that as heterogeneity, we can think of that as symmetry breaking. So we can think about the C. elegans endoderm and how that's specified by a cascade of transcription factors operating sequentially. Identifying the principles of gene regulation has been a central endeavor in developmental biology. In this paper, they use single-cell RNA-seq to identify the transcriptomes of each of the cells up to the 102 cell stage. We find that homeodomain genes are expressed in stripes along the anterior-posterior axis as early as the 28 cell stage. Interestingly, each founder cell lineage, A, B, MS, C, and E, establishes its own regionalization code. And so AB, of course, includes a number of cell types. MS, C, and E are all very specific to specific cell types, to organs. So we're going to see, this obviously, they claim that this establishes a regionalization code. So we'll see what that looks like. These Drosophila-like stripe patterns suggest a deep homology and sulfate specification between the embryogenesis of non-segmented and segmented animals, despite differences in synticum-based and cell cleavage-based modes of development. So synticum-based are what you see in Drosophila. Cell cleavage-based is like what you see in C. elegans. So if we look at figure 1, we can see in A the lineage tree. And we can see that we start with 2 cells on each pole, AB, which is anterior, P1, which is posterior. And then we go down to our different division events. We go down to the 102 cell stage, and we go down even further, but this kind of shows sort of at the end, late gastrulation here. And we can see down here that we have these different sublineages, and then the type of tissue that's formed from that, or the cell types that form from these different sublineages. So in AB, we have neural cells, we have hypodermis cells, we have pharynx, and blast cells. And then MS, we have pharynx cells and muscle cells. E, we have all gut cells. C, we have hypodermis and neural cells. D, we have all muscle cells. And then P, we have germ cells. So this is where we have the sort of the germ cells that are segregated out very early in the lineage tree, and they're kept separate from the rest of the cells. So basically, AB is very heterogeneous. We have different cell types here, forming MS, E, C, and D are all more or less, specific to different, 1 or 2 different cell types. And so we see all that happening either at the middle of the anatomy or towards the posterior end of the anatomy, which is interesting because that gives us, of course, a place for those organs to form. But a lot of this sort of thing happens early on in gastrulation. So we're talking about, again, like from 28 to 102 in this part of the cell lineage. And of course, these symmetry-breaking events are important for setting all of this up. Now, in this example B, we have a t-SNE plot. So we're taking t-SNE1 versus t-SNE2, and we're looking at the different cell types in the different sublineages. So we have cell number. This is color-coded by cell number. So this is where we're going from like small numbers of cells here to larger numbers of cells here. It looks like the larger number of cells are kind of on the outer edges of this, and the smaller number of cells are in the middle. So we have, of course, the germ cells here, the gut cells, they form a group, muscle cells are out here, a pharynx is over here, blast cells are up here, and the neural cells are here. And so you can see that there is this sort of difference in this t-SNE plot of different cell types, and looking at sort of those, what that looks like using dimensionality reduction. And so if we look at this a little bit more closely by sublineage and by gene, we find that MS expresses CEH51 here. E expresses ELT7 here. These are the markers that they're using for these different lineages. So we have CEH51 for MS, ELT7 for E, PEL1 for C, PES1 for D, and NLS2 for P. And so you can see that, these markers are sort of in the neighborhood. They're where we expect them to be, more or less. And then for our fate, we have different cell fates: pharynx, muscle, hypodermis, neural, and blast. And then we have markers for these. So we have FA4 for pharynx, HND1 for muscle, ELT1 for hypodermis, CH58 for neural, and UNC6 for blast. And so they're all kind of where we expect them to be as well. This is telling us the level of gene expression. So this is where these markers are highly expressed on this T-SNE map versus where they're not, which is gray. And so if we look at their results, we found that the cells follow developmental trajectories according to their founder cell origins, each identified by the expression of genes known to be expressed in a lineage-specific manner. So these markers are expressed above sort of the control level. So when we have differential gene expression, we look at some housekeeping gene, and then we look at the expression of this marker above sort of background, or chance, basically. So the higher the level of expression, the greater the fold expression, the more strongly that marker is expressed, and the better it is for using it for a marker. And if they're in cells that we know are that type of cell, then that means that it's a good marker to use. As observed by other studies, the founder cell lineage trajectories are oriented according to cell fates, with similar cell fates converging between lineages. So, for example, the cells of the ectomesoderm producing C lineage formed by a bilobed cloud with the muscle part adjacent to that of MS and D lineages. Similarly, MS cells that will form the pharynx orient towards the pharynx producing portion of the AB lineage. So figure 2 is where they try to infer the transcriptome of these different cell lineages. And so here, I think D is a good example of what this looks like. In D, we see that we kind of recapitulate that the lineage tree. We have different markers for each cell. So A, B, and P1, we use MEX3 as a marker, and they're both expressing MEX3 at chance or at background. And everything that's dark blue is expressed at background. Everything that's a lighter shade leading up to red is expressed more highly. So red is high expression, dark blue is low expression, and everything in between red and blue, the lighter colors, are moderate amounts of expression. So we see nothing in AB and P1 for MEX3. When we get down to the 4-cell stage, we begin to see differential expression of SMA9 in ABA, FLH3 in ABP, EMS has PES10 and SMA9, and then P2 has AMA1. So, but you can see what happens here. You start to get different things expressed differentially in different lineages or sublineages. So ABA and ABP, despite originating from the same mother cell, are expressing different markers differentially. EMS and P2, descending from P1, do the same thing. This gets even more pronounced as we move down the lineage tree to the 8-cell stage, where we have a bunch of genes here, and we have differential expression. Now that's, I think, a lot more clear, where ABAAL and ABAAR basically are not expressing anything differentially. In ABPL and ABPR, you have HLH26 and HMG4, which are maybe expressed a little bit differentially, but not very much. But then when you get to MS and E, they start expressing, TBX35 in MS and END3 in E are expressed at a much higher level. And then in the germline here, P3, and then in C, which is not the germline, you get different, like MOM2 in C, and then CY3 in P3. So you get these different types of differential expression in different sublineages. And this just becomes more pronounced as we move down, especially when we start to get to the 28 cell stage, where we start to get like these high levels of differential expression in different markers, and even in closely related lineages. But in overall, we have these groups of cells here, like ALA and ARA, that are much more similar than things like PRA and PRP in this other sublineage. So you see, as you move away from each other in terms of it being part of the same sublineage, you start to express different things differentially. So you see here MSAAA and MSAAP and MSPPA and MSPPP. They both actually express the same things differentially, which is interesting. And it's different than some of these other daughter cell combinations where they're expressing other things differently. So it's interesting that there's some cases where things maybe get preserved or things aren't that much different, and places where things are sort of always unique. And so what this might suggest is that you have a specific cell type that's sort of representative of this lineage. We know, like from what we just said before, that in these lineages like MS and E, that you're going to have like one or two cell types produced as opposed to a mosaic of cells in the AB lineages. So this is kind of what they're getting at here. And then, as you go down, it gets even more unique. But you do get patterns across sublineages. Maybe this is where you start to coordinate organ formation and things like that. So in figure 3, they start getting into how all this plays out spatiotemporally, because so far we've looked at the temporal domain, we've looked at the lineage tree, and we've seen that there are differences as the lineage tree unfolds. That's obviously in space, but there's also the spatiotemporal aspect, which is where we have these different lineages that are going to be sort of located in a certain location in space, and then there is overlap in some of these. And so we want to see like these because in Drosophila, when you get overlap of gradients, they sort of form, this is the place where they form stripes. So if you get two gradients that overlap, they form a sharp boundary and you get stripes. So what happens in C. elegans? Well, we'll look at two different cells here. ABPL, PPP, and EALA. So these are in different parts of the lineage tree. So we actually have this little graph where it shows kind of these different classes of genes that are found. So for ABPL, PPP, we have 921 genes. We have this blue area, which are the homeobox domain genes, 103 genes. We have transcription factors expressed in the cell of interest, which is here, and then Gatta-Zinc finger transcription factors, 14 genes. So we have the same thing for each cell type. And then here in this yellow, we have this enrichment, or actually it's the overlap between the blue and the yellow. It's this overlap area. And this overlap area is enrichment for homeobox domain transcription factors, and this is the case at a significant p-value, and this is true for 7 genes. So 7 genes are enriched for homeobox domain transcription factors. For EALA, we have both enrichment and depletion. So what we're going to have in some of these cells is we're going to have things that are enriched. We're going to have things that are upregulated. And we expect to see that because that's what's going to drive differentiation between cells. We also have, in EALA particularly, is this depletion of cells, or this depletion of genes. So we have things that are kind of shut down and things that are turned up. And so this, in the case of EALA, we have enrichment and depletion. So overlap between the red and the yellow, that's enrichment for gamma zinc finger transcription factors at a significant p-value, that's 5 genes, and then depletion for homeobox domain transcription factors rather than enrichment is at a statistical significance of 0.01, and that's 2 genes. So you see the logic of this test. We're looking for the overlap between transcription factors expressed in the cell of interest, the homeobox domain genes, and then if it's enriched or depleted. The same thing for gamma zinc fingers and transcription factors expressed in the cell of interest, and that's either enriched or depleted. So if we look down at this these graphs in B and C, we see on the left homeobox domain transcription factors, and on the right gamma zinc finger transcription factors. And so we see enrichment in red and depletion in blue. We can see that for gamma zinc fingers, almost everything is enriched in this part of the lineage tree from the 4-cell to 102-cell stage. And then we see for homeobox domain transcription factors, we see a lot of enrichment in the AB lineage and their sublineages. But we also see a lot of depletion in the E, C, D, and P sublineages. So we actually see this with respect to these different, so homeobox domain transcription factors, of course, were this area here. And we can see that for homeobox domain transcription factors, they're both enriched in AB and MS sublineages and depleted in E, C, D, and P sublineages, meaning that, of course, there are going to be differences in differentiation here between these cells eventually. But also that there's a spatial segregation, that towards the posterior end, you get this depletion of homeobox domain transcription factors. We also see for gamma zinc finger transcription factors no such spatial patterning, although we see enrichment pretty solidly across the E sublineage, which means that whatever cell types are going to come out of the E sublineage are going to be enriched for gamma zinc finger transcription factors. And so in this graph in D, we have the different stages. We have the lineage for at different stages. So this is 15, 28, 51, and 102. And then the dark blue is AB, and everything goes out to MS, E, C, D, and P. So you can see the sort of the color coding here. So each one of these columns here go down for each one of these classes of transcription factor, and we want to see if it's either depleted or enriched. So for helix loop helix DNA binding domain, most of the time it's enriched, although it stops being as enriched as it is in the early stages later. We see a little bit of depletion in, I believe it's the P lineage in the 28-cell stage, which is interesting, but we don't really know too much about that. The homeobox domain, however, has sort of patterns of depletion and enrichment as we go along, with a lot of depletion in the E sublineage, as we talked about in the last figure. And so this just shows that this depletion is established actually somewhat early in development. The zinc finger C2H2 type is slightly enriched across all times and across all sublineages. You see for zinc finger C4 type, you get a lot of depletion for ligand binding domains of nuclear hormone receptor. We also get a lot of depletion and enrichment. A BZIP transcription factor is almost always slightly enriched. Forca domain is also almost always slightly enriched. Gamma zinc finger is almost always slightly enriched, and so on and so forth. So we can see for each of these categories of transcription factor that there are these different patterns in different sublineages at different stages of development. So we have the spatiotemporal diversity here of these different transcription factor families. Then we can see this broken out by sublineage. So this is the MS sublineage, the E sublineage, the P sublineage, And this is true for both cell stage and for sublineage. Finally, if we take a look at our markers across the embryo, so remember the lineage tree goes from anterior end to posterior end. You have these different cell stages, the 28 cell stage, the 51 cell stage, and the 102 cell stage. And you can actually look for spatial expression of these different transcription or these different genes in different cells. So actually what we see here is we see a lot of expression across the embryo for different, so actually within the AB lineage here, we see the gene expression matching the AB lineage almost exclusively through, and then we can look at the MS lineage and the C lineage, and we can also get a signal. So what we have are these lineage-specific stripes across the early embryo throughout gastrulation. Just like what we see in Drosophila, resembles Drosophila. It's not exactly like Drosophila because we have a different mode of division and expression of these genes. Okay, now this is a fun paper. I found this paper when just looking through the literature. This is an older paper. This is from 2017, and this is from Molecular Biology and Evolution. The title of this paper is Multiple Regulatory Modules Are Required for Scale-to-Feather Conversion, and it's actually a paleontological paper, but it's also an evo-devo paper. So it's evolution of development, but they're looking back into evolutionary history and looking at how we get these genetic regulatory modules that control scale production and feather production, and especially in lineages where we go from scales to feathers or feathers to scales. So the abstract reads, the origin of feathers is an important question in evo-devo studies, with the eventual evolution of vaned feathers, which are aerodynamic, allowing feathered dinosaurs and early birds to fly and venture into new ecological niches. Studying how feathers and scales are developmentally specified provides insight into how a new organ may evolve. We identified feather-associated genes using genomic analyses. The candidate genes were tested by expressing them in chicken and alligator scale-forming regions. So this is where they're looking at these genes, they're trying to find things that are associated with feathers, and then they want to express them in certain scale-forming regions that will allow us to do experiments where we can see their effect on the phenotype. Ectopic expression of these genes induce intermediate morphotypes between scales and feathers, which reveal several major morphogenetic events along this path. So we're looking for the things that form scales, the things that form feathers, and if we can form something in between those two, and indeed we can. So there's several major morphogenetic events that we need to be, sort of figure out how this process works. So the first is localized zone growth formation. The second is follicle invagination. The third is epithelial branching. The fourth is feather keratin differentiation. And the fifth is dermal papilla formation. So we have this 5-step process. And if it's disrupted in different ways, we either get scales or feathers. In addition to molecules known to induce feathers on scales, such as retinoic acid and beta-catenin, we identify novel scale feather converters, SOX2, ZIC1, GREM1, SPRY2, SOX18. So there's some SOX genes in here, a ZIC gene, and we know those are important in developmental pattern formation and cell differentiation. So we get these scale to feather converters, and we get this induction of feathers on scales. These novel scale feather converters induce one or more regulatory modules guiding these morphogenetic events. Some morphotypes resemble filamentous appendages found in feathered dinosaur fossils, whereas others exhibit characteristics of modern avian feathers. We propose that these morphoregulatory modules were used to diversify archosaur scales and to initiate feather evolution. This goes back to dinosaurs and how, we can think about the common ancestry of some of the modern lineages and clades that have feathers and have scales. The regulatory combination and hierarchical integration may have led to the formation of extant feathered forms. Our study highlights the importance of integrating discoveries between developmental biology and paleontology. So, of course, in amniotes, we have different types of these intragumentary appendages, which involve scales, feathers, and hair. So you'll see pictures of the feather follicles. They look like hair follicles, and they are also involved in forming scales. These appendages exhibit region-specific distributions on the body surface. So, for instance, many birds have scutate and reticulate scales on their lower legs and feet, and have feathers on most of the remaining body parts. Crocodilians have different types of scales on their body surface. So this involves avians, this involves lizards and amphibians, this involves snakes, and this also involves other amniotes as well. The feather is a novel organ that evolved from dinosaur integuments. Developmental biology studies and recent fossil finds reveal that feathers evolved from a series of novel morphogenetic events, which we talked about in the abstract. And they also talk about some of the early epidermis-dermis recombination experiments, where E9 shaped dermis does not alter the fate of feather epidermis. However, at a later stage of development, which is E12, shaped dermis can cause feather epidermis to form some scale-like skin appendages. So these are some early studies by Rawls in 1963 that look to understand sort of the developmental regulation of these different structures. So this is an example. This is a phylogeny of amniotes. So this is the stem amniotes here, which is the common ancestor of all amniotes. We have mammals here, avians, and non-avian reptiles. So we have these three different groups. And so in mammals, we have hair, of course. We have the hair follicle. We have bulge stem cells. We have these other structures. Then if we look over to avians, we have feathers and scales. We have feathers. And again, they form this follicle. They form this invagination into the skin, just like hair. So the epidermal invagination is here in hair and here in feathers. So the feather is wider, and so it's a bigger invagination. You have these stem cells here, and you have the supporting structures around it. Then you have scales, and the scales are like these sort of bends in the epidermis of different shapes. And so you see that here. You see that also in non-avian reptiles, where you see maybe a little bit broader diversity of scale types, but you have these other scale types that are conserved, I guess, between avians and non-avian reptiles. So that's what we're talking about. We're talking about hair, we're talking about feather and scale development together in the same group of organisms, and then we have just exclusively scale production in non-avian reptiles. But they all have this sort of common origin in stem amniotes. Then we have this image B here, which is upregulated genes in the feather region. So they have these different regions that they pick out in this embryo, 1, 2, 3, and 4, and they kind of zoom in on that area. They look at the different types of genes that are involved in the formation of different things. So they have this feather versus scale, E9 leg, E9 wing, and then E9 dorsal. So 1 is dorsal, this red box. This is feather versus scale. The blue box 2 is leg. The box 3 is wing, and then box 4 is down here. And they just mark this off, and they do this sort of Venn diagram that shows that there are these genes sort of that are expressed in common. This leads us to this comparison where we have between upregulated genes in the feather region, not upregulated genes in feather development, between this region here on the dorsal side of the embryo and the scutate. And then we have this comparison here where we have 218 feather region independent genes, 552 feather development dependent genes, and then 102 feather associated genes. So that's how they get to feather associated genes. Here's another figure that shows the expression of ZIC1 in an E9 embryo. So you have ZIC1 in these different areas. You don't really see a lot of expression. A little bit down here towards this tail, you see some sort of patterning in different parts of the embryo, but not very strongly, as opposed to SOX18, where you see strong patterning at the same developmental period in different parts of the embryo. We also see this analysis where we have feather at E7 and E9, and those genes are upregulated compared to the scale genes, which are not upregulated. And this is their genomic position up here, and then this just shows their expression level. We see this for SOX18, but in SOX18 we have feather-related expression here, and then scale-related expression. So we get some scale-related expression for SOX18, but a lot more feather-related expression. Then we have these in situ hybridizations on the right. So this is where we have the control, which is here, SOX2, GRAM1, SPRY2, beta-catenin. So these are all the different markers that they're using. And then they show the thigh and the shank. So this red box is the thigh. This blue box is the shank for each sample. So the shank is down here. The thigh is up here. This is the thigh. This is the shank. You don't see very much control. You don't really see any expression of anything in the control, obviously. SOX2, you start to get some expression. Here you see this patterned expression, but you don't see anything in the shank really, but you see it in the thigh. And of course, they have an inset that shows the scales forming here. You have GRAM1, which forms this different type of scale here. And you see that in the thigh, but not the shank. SPRY2, you get this scale formation here where you have in the thigh, that's a different pattern. And then in the shank, you don't see anything. Finally, beta-catenin, you see this pattern here where you have strong expression in the thigh, but also in the shank. And you see what that looks like in the inset on the shank in terms of morphology. And so these red arrows, these are feather bud mesenchyme cells. And so for SOX2, GRAM1, SPRY2, and beta-catenin, you have these feather bud mesenchyme cells in this structure. So finally, what we end up with is the ability to form intermediate phenotypes from molecular and chemical perturbations. Their data suggests that a successful formation of feathers requires the integration and coordination of these molecular modules. In this work, our developmental biology-based approach revealed molecules that are involved in each regulatory module. Our studies provide groundwork towards an understanding of how these regulatory modules are integrated at the molecular level for the generation of real feathers as defined by the 5 criteria. And so the 5 criterion are these 5 steps that they mentioned in the abstract. Okay, so Lilith and I had a discussion in today's meeting, and our discussion was about the difference between artificial neural networks and biological neural networks. And I had mentioned that we had published a paper back in 2021 on the topic, we being myself, Krishna Katyal, and Jesse Parent. And the name of this paper is Connectionism, Complexity in Living Systems, a comparison of artificial and biological neural networks. So this is on the arXiv. You can see here, this is the stub. And in this paper, we try to have a discussion on some of the major differences between artificial neural networks and biological neural networks. Nothing earth-shaking, really. We kind of characterize the different, the major differences between the two different types of systems. This is something that we did a presentation on one of the early Neuromatch conferences, and then we decided to publish this. So this is the preprint. We make a number of comparisons between artificial and biological neural networks. So we have this part of the introduction where we talk about, we don't really get into bio-inspired models or more exotic types of models. We just focus on artificial neural networks, different types of things that artificial neural networks that can do, which seem to be, lifelike or cognitive. And so, we ask the question, okay, what are the differences between models like that and biological neural networks, knowing that artificial neural networks are not going to give you the same result? So maybe what are some of the reasons why that difference exists? And so we kind of get into some of the points here. The first point is that there's a sparse behavioral output and internal representation in artificial networks. So a sparse behavior output means that it doesn't provide a rich repertoire of behaviors. It provides maybe one stereotype type of behavior or multiple stereotype types of behavior. And those types of behavior are generally the outcome of training the model. So you have to train the model before you can get an output. In biological neural networks, of course, you have learning and you have development. But some of what a biological neural network does, especially for stereotype types of behaviors, are innate. They're born of internal to the organism. So, we have organisms with innate behaviors. We have organisms with autonomous behaviors. And we don't see that in artificial neural networks. They also have a difference in terms of representation. So artificial neural networks have sparse representations compared to behavioral or compared to biological neural networks. And so we can think of this as a difference in representational complexity. So this is an example of the difference. In biological neural networks, we have a number of different cell types, a number of different communication paths, and other types of support mechanisms that make for this biological system. And this is a picture of a couple of neurons connected by dendrites and axons and synapses. And so all of those things are there. Plus we have support cells like glia and astrocytes, which keep everything as a homeostatic system. In the inset, we see this artificial neural network, which is basically weights or computational weights and a cell body that sums those weights and provides an output function. So we don't really have that same type of complexity here. We don't have different levels of complexity like biological networks. We have a molecular level of complexity. We have a phenotypic level of complexity. We have a network level of complexity. Here we just basically have the network level of complexity and some phenotypic complexity if you think that the cells or the units or the connections are, sort of stand on their own. Another thing we talk about is heterogeneity, and specifically that biological neural networks are heterogeneous. And so, as in an artificial neural network, basically you have undifferentiated units, and those units are connected by undifferentiated weights, and they combine in a combinatorial manner to give you the complexity that you see. So you can model a dynamical system using an artificial neural network just fine, but it doesn't offer the sort of the functional heterogeneity of a biological neural network. And so we just assume that that functional heterogeneity plays some role in producing better outputs, if you want to think of it that way, than artificial neural networks. So in biological neural networks, we see great heterogeneity. If we go back up to figure 1, we see that there are different cell types. We have different types of structures. We have different types of relationships between cells, different communication channels. Heterogeneity is the rule rather than the exception. But in artificial neural networks, we tend to want to systematize everything and make it homogeneous. Again, we don't talk too much about bio-inspired systems. In bio-inspired systems, we might think about this a little bit differently. But in this case, we're just kind of drawing out this distinction. So another difference between biological neural networks and artificial neural networks is this difference between something like a complex brain network or a complex network and a network that is sort of organized either hierarchically or by layers. And so in artificial neural networks, we have layers, and in deep learning networks, we have many layers, and it's just a matter of adding layers. In a complex brain network, however, we play off of this heterogeneity. So we have different processing centers, but we also have a lot of interaction between, say, modules or processing centers or regions, and they follow the laws of a complex network. So you see hubs emerge, which are centers of processing that can emerge from other types of topological organization. You see other types of like sparse connectivity versus rich connectivity in different regions of the network. So you think about the brain, some parts of the brain are, when I have a task in front of me, some parts of my brain are more active, some parts of my brain are less active. We don't have any of those distinctions in artificial neural networks. We also talk about robust function, which is, of course, an offshoot of these other things we've been talking about. And we use the example of opponent processes being theory of function in vision in mammals. And so we talk about that a little bit. We also talk about fault tolerance and robust function more generally. And so this is an example here, again, of this type of heterogeneity, where we have many different types of neuronal morphologies. They're all connected together. They're connected in ways that aren't sort of strictly in unidirectional or even hierarchical. So then we kind of consider, why should we emulate biological neural networks at all? Obviously, we're giving sort of a comparison between artificial and biological neural networks. And, that's fine if there's just a simple comparison. Obviously, they're different. But perhaps we want to actually not emulate biological neural networks because they might not be the best way to do things we want to do in a computer. And that argument certainly exists. But of course, people have found biological networks attractive. They've found at least the metaphor of biological inspiration or biological, sort of the goal of biology, emulating biology to be the thing to do. So that's where we're kind of going with this. We kind of think of the things that we're trying to do with, that we're trying to do with both types of networks. So we think about this sort of trade-off between interactive and independent parallelism. How can both types of networks manage that? We also think of things like goal-directedness as outputs. And we think about the information processing capabilities of biological systems versus the biological processing capabilities of artificial systems. And I think we talk about this in the paper, there's some energy consumption estimates So for biological networks, they can do a lot more processing with a lot less energy. And of course, artificial neural networks are notorious for gobbling up energy and doing less than biological networks. Then we have this section on artificial neural networks as a phenotype for data. So this is where we might want to process data using an artificial neural network. But because it's not embodied, we might not have the kinds of, the kind of power that we have in a biological neural network. So one thing to think about is, what if we embodied an artificial neural network? If we embodied an artificial neural network, would that allow us to process the data better? Or, maybe create a context for the data? Maybe create a situation where there's some spatiality or spatial heterogeneity to the data? Or at least the analysis? It's something to think about. So this is an image here, figure 3, where we have this cartoon organism where we embed an ANN, and it's anatomically salient. It has some sort of anatomical realism to it. And so that's kind of our thought experiment for the rest of the paper. And so that's all we have for this. Now, this is something that we're going to be adding to, and I would like to think about this with Lilith. What could we kind of work on as a project here? What could we kind of sort of follow up on? I think he mentioned that he was interested in the energetics question. Perhaps some of the other questions other people can take on if they see fit. If you're interested, let us know.